Welcome everybody to the latest edition of the REPROCELL Webinar series. My name is Bob Anand. I'm Technical Product Manager here at REPROCELL. I'd like to introduce our speaker today is Dr. Daniel Haag from Newsight. He's going to talk about Newsight's SendFire technology and its use in neural networks. I would just remind everybody that if you have any questions during the presentation, you can use the Q&A feature located at the bottom of the webinar window, and we'll respond to your questions at the end of the webinar. Dr. Haag?
Yeah, thank you, Bob. Thanks everybody for joining in. I would like today to give you some background on our technology for making neurons from human pluripotent stem cells, and particularly on the background on the characterization of these cells, and show you some application examples for neurotoxicity testing and CNS liability assessment. So there are obviously different ways of turning an iPS cell into a neural cell or into a neuron. And the most common approach used so far is, or uses protocols that apply stepwise differentiation through extracellular signaling cues like growth factors and patterning molecules, which basically recapitulates early embryonal development. And so this can be done in a 2D format or in a 3D format, and both approaches have their own advantages and disadvantages. For example, they have very sophisticated 2D protocols for really nice specification of neuronal subtypes. And on the other hand, the 3D organoids or spheroid conformation basically gives you a more connectivity, and then you have the tissue structure maintained in that, and also a more physiological composition of the different cell types that you might use for your assay. So, however, Newsight is taking a fundamentally different approach on that. So we're not going through a stepwise differentiating process, but we are rather directly converting the identity of an iPS cell into the identity of a neuronal cell or a glial cell by exposing iPS cells or stem cells to neurogenic transcription factors. And this has a couple of advantages. One of the largest advantages is actually that you end up with a very homogeneous population of a certain identity, depending on the different combination of the transcription factors used. And also that this population that you end up with, that you produce, is for each of the individual cells pretty much on the same maturation stage. So whereas in the all stepwise differentiation protocols, you always have some more or less differentiated cells, some late stage progenitor cells, and already some neurons that have capabilities of firing. Whereas with this approach, with a direct reprogramming approach, you pretty much have every neuron at the same stage of maturation, because the transcription factor applies a dominant stimulus overwriting the existing transcriptional programs, and therefore leading to a, I would say, synchronized, but cell cycle independent, more synchronized maturation stages. And also this process itself speeds up the maturation to achieve functional neuronal identity. And so this approach also harbors some major advantages, particularly inside of assay development, as you can produce those different neuronal cell types separately, and you can also modulate and modify those cell types separately. You can introduce a certain reporter or a certain mutation in a certain subtype or in a fraction of the subtypes, and then you can mix and match those different types to generate highly functional co-cultures. Where on that you can build your assays depending on the endpoint readouts that you are interested in. And so applications of that is, for example, neurotoxicity screening, where we present an example or two examples about this, as well as phenotypic drug discovery or efficacy testing of compounds that you're interested in. So our standard cultures that we are using at Newsight, go to the next slide. 1 second. Here we go. Okay, so the standard cultures that we are using here, and where all the data that I'm going to present here are derived from, consist of glutamatergic excitatory cell type and a GABAergic inhibitory cell type in a ratio of 70:30 on a bed of primary human astrocytes. And this exact configuration we are also providing under the brand name Synfire co-cultures. And so this is the standard culture where we have most of the characterization work done on, and which we have most of our assays established on. And these co-cultures in that configuration are already suitable for a couple of assay readouts, such as immunocytochemistry, or as you see here, an example for live cell imaging, where you can track in real time the neurite outgrow of different cell types, as well as microelectrode applications for assessing neural network activity, and we will hear definitely more about this. And lately, we have been testing calcium imaging on these standard co-cultures of the 70-30 ratio. So regarding the characterization of the neuronal cell types that we are generating with this approach, the most important part, of course, is that they do what a neuron is supposed to do, that they're electrically active and they fire when they're supposed to fire. And using patch clamp analysis, we extensively characterize the properties of those different neuronal cell types that we generate. And we can, for example, see principal neuronal physiological properties of a neuron, such as voltage-dependent potassium and sodium currents, but also a little bit more advanced, like spontaneous bursting events, like a train of multiple action potentials occurring. Of course, we have evoked and spontaneous action potentials firing. That's very, very basic property of a neuron. And also we see in our cultures very strong postsynaptic currents indicating the presence of functional synapses. And then, as well as already indicated, that this technology of the direct reprogramming speeds up the maturation process in those cells. The cell intrinsic properties that would reflect those of a mature in vivo neuron, such as, for example, the resting membrane potential, is something that we reach already between week 4 and week 5 at a stage of -60 millivolts. So this is indicating for us this is really close to a very mature neuron in vivo. And also very important is that the cell types that we're generating are indeed those or pure, consisting of those that we expect them to be. So in this case, if we co-culture only the excitatory glutamatergic neurons, we can only measure excitatory postsynaptic currents that we can completely block by AMPA antagonists. And if we only co-culture the inhibitory neurons, we see the same for inhibitory postsynaptic currents that we can then block with GABA antagonists such as picrotoxin. Besides GABA and AMPA, we have also robust NMDA currents that we can measure in the mature neurons, as well as GABA, tonic GABA currents. So we have NMDA currents, synaptic NMDA currents, as well as persistent currents, extrasynaptic currents. And here's an example of tonic GABA currents. And we will address the synaptic transmission a little bit later. But so this indicates this is a very mature functional neuron where you can run a lot of functional assays on. So besides the electrophysiological activity, of course, it's important for us, and especially for application of more high throughput assays, to have some characterization and background knowledge on the structural and morphological properties of these cells. And we can, for example, see that the co-culture system as it is has a nice distribution of GABAergic cells in between the standard neurons, or in between the excitatory neurons, and as well the glia cells are very nicely distributed, not forming any clusters, clumps, or balls. And we see synapse staining, very dense synapse staining, and formation of networks, and especially important, the establishment and expression of endogenous neurogenic transcription factors like NUN in this case. And especially important for high content analysis is that we can, in a more mature stage of those co-cultures, we can also analyze fine structures like spines. So in this case, this is one single labeled neuron, genetically labeled neuron, on a dense bed of unlabeled neurons, so that you have all the environment in there that it needs to turn its filopodia into spines and so on. So all the markers are expressed, and the morphology is neuronal. And with this approach, we already have a good approach. We already have a good angle on making a stable product or a stable cell type for assay application. But of course, there's a little bit more to that if you really want to go for in vitro assays, especially if you are from a cell type that has never been actually really grown in vivo. And so this is a little bit of the boring part, but we found that this is extremely important. So all of our production pipelines undergo a very, very tight QC process, which starts obviously with the iPS cells, with the starting material, which involves exome sequencing of the iPS cells, copy number variations, karyotype, pluripotency assessment, and so on for each bank. So every 5 passages, basically, we have to do all this analysis again. And also the IM batches that we produce from these QC'd iPS banks have to undergo very tight quality controls and quality assurance processes based on patch clamping, electrophysiological assessment of cell intrinsic properties and firing properties, but also on network formation on microelectrode arrays, and especially in response to certain tool compounds to see if the connectivity and the responsiveness of the networks that are forming are what we are expecting to receive on that. And indeed, following all these QC processes and establishing this pipeline, we indeed see very low variability across IN production batches, and we also see comparable, very comparable functionality and activity of IN production batches between different individuals or different iPS lines from different individuals, which also then leads to network development in a very tight margin from batch to batch. So basically that you always know that your assay time point needs to be at a certain level of synchrony or network activity, and basically we can with these QC can guarantee that you will always have, after 23 to 25 days, you will have the synchronization of your network occurring in that. And ultimately these luckily leads to a very low number of outlier replicates on your assay, which basically cuts down on the cost on your end or on the user end. So what can you do with those cells or with this approach? So a very nice example that is more on the image-based side and that we have been testing out recently is genetically labeling of the different subtypes of cells that we produce, excitatory in one color, inhibitory in the other color, and spike in those in a dense bed of excitatory, inhibitory astroglial cells, so that the environment and the signaling molecules is completely as you would expect it in a real dense grown brain, but you can still track a single neuron specific to a certain neuronal subtype in live behavior. And so just application here for live cell imaging, where we have excitatory and inhibitory cells, separate channels tracked over time under the chemical challenge of lantrocolin A, so a compound that disrupts actin polymerization and therefore affects cytoskeleton. And then we see in both cell types, we can separately track different degrees of effects on the neurite outgrow. So this is just one example for a live cell imaging assay, which is that confirmation, not really high throughput assay, but of course you can also apply the same inhibitory, excitatory, astroglial cell types or culture types on a more high throughput approach. And something that we have been recently developing is the transduction with encoded synapse labels like postsynaptic and presynaptic proteins carrying labels that we can basically transduce to either a certain cell type or to a fraction of a cell type, and then again can read out the formation of synapses or the abundance, the relative abundance in response to a stimulus or to a disease trigger in real time. But of course you can also use the same cell cultures and fix them and then stain them with antibodies and do all the structural analysis as usual, basically. But for us, the most important and most informative assay so far is actually electrophysiologically based. So in this case, we're using 48 well microelectrodes from Axion, where we seed our standard culture, 70-30 excitatory inhibitory plus astroglial cells on the grid of electrodes. And we can then measure from the whole population not only the single activity or single spiking of a neuron, but the whole network formation. So basically how the neurons fire and wire together. And so this network activity, this synchronization or this coordination of activity can basically be regarded as the functional integration of different neuronal properties, such as excitability, synaptic competence, and so on. And with the quantification of parameters that describe this network structure or this network activity, we can gain a lot of information of what is going on activity-wise in those different cultures or upon a certain challenge. So, for example, we can collect with a single recording measurement, we can collect over 30 parameters that describe how a burst is changing over time or upon the challenge, the burst duration, how synchronized these cells are, how they are interacting together, how much of this spontaneous activity is actually occurring within a burst or outside of a burst. So all this can be used to basically generate neuroactivity profile of the cells under different conditions. And we found that our Synfire co-culture cells are actually particularly suited for that, since they rapidly mature and therefore rapidly produce these synchronized neuronal networks. So starting at week 3, we see the first synchronizing network burst, and by week 4, we have a very nice regular pattern of coordinated network activity. So this is very great, and then gives you a lot of information already. However, you have to know about network activity, so that coordination of neuronal firing can occur through chemical synapses, but it can also occur through electrical coupling, which is actually the first way of connecting or coordinating neurons in neuronal development. So in early cortical excitatory cells connect by gap junctions and by electrical coupling, and only this later in developmental stages is replaced by chemical synapses. And a nice paper last year actually showed that iPS-derived neuronal cultures that show synchronized network activity derived by a conventional stepwise differentiation recapitulate this early type of synchronization. And if you apply a gap junction inhibitor that blocks the electrical coupling, you can actually decrease the frequency of this network bursts. Like this is the shift of this darker purple curve here. And so depending on, of course, what your assay is supposed to be used for, if you want to study very early developmental effects, this might be something that you want to look at. But usually when you want to test a certain liability or a toxicity, you want to see the effect of a mature brain or of a mature neuronal tissue. And so therefore we were testing our cells, since we have this nice network activity, if that is also driven through gap junction. And actually we don't see any impairment of any of the parameters that describe network function, network structure by application of carbenoxolone. And even more when we challenge the system by removing the GABA components, the inhibitory GABA component, by blocking GABA receptors, we see usually hyperactivity, and we can see that this hyperactivity is absolutely not affected by blocking gap junctions, indicating that indeed this activity is mediated by chemical synapses. So this is a small but very important step in making conclusions out of your results that you get from an assay that you run, if this is chemically synapse driven or if this electrical coupling driven. So this is an important characterization step that you have to undergo. But besides this functional characterization and structural characterization so far, we also attempted a more molecular characterization approach, obviously. And for this, we did Rna-Seq analysis for the whole culture as it comes in the standard configuration with the 70-30 plus the astrocytes at different time points of maturation. So in this case, for 6 weeks, for each one time point per week. And yeah, we see basically a broad representation of neuronal ion channels, sodium and potassium channels, as well as a very broad representation of neuronal signaling pathways through different receptor pathways. And so keep in mind, so this is the bulk culture. So basically this is not separated between excitatory and inhibitory neurons. This is currently ongoing, but this data is until not yet available. However, this data is of course available upon request, so we are happy to share this information upon request, and don't need to read through all the tables here, but just shoot us an email and then we can send you some information about this, what receptors are expressed. But of course, expression is not the only thing. They need to be functional to be suitable for testing it. And therefore we conducted a Pharmaco Profiling project on those standard co-couches on the Sunfire 7030, standard co-couches using two compounds as agonists and antagonists to the receptors and receptor subunits or subtypes, isoforms that we see being expressed at the different time points. So again, this is also a work in progress, but so far we have one time point assessed, so time point which is usually used for most of the assay applications, but we're in the process of doing all the other time points which can give you information about when you would basically try to assess an NMDA pathway versus kinet assay stimulation. So I don't want to go through all of these compounds, just as a quick example how we approach this. So we use patch clamp analysis to first see if the receptor is actually functional, and in this case here for GABA A receptors, we see muscimol as an agonist, indeed producing a current which can be blocked by the antagonist pyridoxine. But also we see that this receptor signaling is functionally integrated into the network activity, as we see a dose-dependent decrease in the activity by applying the inhibitory agonist, and we see in the other way around when we block the inhibitory component of that network. It's a little bit more difficult or complicated for glutamatergic receptors or AMPA receptors, although we also see here a strong current induced by the agonist and a blocking of the current by the antagonist. But we see not a clear dose-dependent effect, but we see increase of firing in low to medium doses in network burst and in signal spiking, which is basically reflected by the weighted mid-firing rate. But we see on the other hand, if we block AMPA receptors, we see a dose-dependent decrease in other parameters. This not only shows you that these receptors are functionally integrated, but also that it's a fairly complex integration. And so with all these two compounds that act on different receptor pathways, we can then generate profiles of the different parameters describing the network activity, and could therefore already identify if we see a compound that we don't know which mode of action it is going through, we can match it up with a signature that we know from our two compound profiling. And we already have an idea if this is generating or this is affecting this or that receptor pathway. So, and after having characterized the cells to that level, we were kind of encouraged to go to a real world application. And we started with a set of environmental toxicants that has been described and well established to be neuroactive or neurotoxic, mostly insecticides, as well as negative controls and a positive cytotoxic control. And for now, we reduced the set to mode of actions that we know are reflected in our system, which is GABA A receptor pathway, as well as voltage-sensitive sodium channels. And with this, we did a dosing experiment, and again, the same readout on microelectrode arrays, using all these different parameters to characterize alterations or impairment of the network structure. And for all the compounds that are affecting GABA A receptors, we see what we expected so far already by the dosing with two compounds, that blocking the inhibitory component increases the activity and mostly leads to a increase in the burst duration. So that is already nice. We can identify a neuroactive effect. So compared to negative controls, which are not shown here, which are flat in all the parameters, we see in specific parameters a dose-dependent increase, or at least a response in a certain dose range. When we're looking at the second group of environmental toxicants that we have been testing, it's getting a little bit more complex with the responsiveness. So these are, again, all those that act in different ways on voltage-sensitive sodium channels. And here we have actually two groups. We have periphery type 1 and periphery type 2 compounds, and actually a mixed type here. And we can see that those two, although acting on voltage-sensitive sodium channels, they act in kind of a different way of modulating those channels, those different types of peripheries. We can already distinguish by looking at the different parameters that are informative about our network activity. And we can, for example, see that we have an increase of firing in the type 1 peripheries, but a decrease in the burst duration compared, for example, to dieldrin that acts on GABA receptors in the previous slide. But for the type 2, we see actually a biphasic response in most of the parameters, and as well a reduction in burst duration, but also very strikingly a complete disruption of the network structure, which is then reflected in the complete lack of synchrony. So again, by this, we can already see, looking at the different parameters of the network structure, we could already group and say this compound acts a little bit more like this in this compound where we already identified a profile for. So this is kind of an easy target, since we know already the mode of action for these test set, but of course it's getting more complicated. But what another strength of taking this approach is that we can distinguish or separate structural from functional neurotoxicity or neuroactive effects. In that case, we have plotted the weighting final rate, so the spiking, versus viability measured by cell titer blue and LDH release. And we see for the negative, of course, no change at all. For the cytotoxic oxidative stress compound, we see a decrease in activity, probably due to the cell death that we see. But in all those environmental toxicants, we don't see any effect on cell viability, but again, this striking effect on the neuroactivity, which is very important to separate an acute neurotoxic effect from a long-term or maybe a short-term neuroviability impairment. So again, so this is nice since we have the mode of action ready. But for an in vitro approach for developing a predictive assay for neurotoxicity, you usually don't know what your compound is going to do. And so therefore, not a single in vitro assay cannot cover all of the potential tox pathways, obviously. And so we were in the next steps trying to focus a little bit more on different applicability domains of our assay and characterizing the assay if this is ready to use for that. And the first type of seizure liability, first type of CNS liability or neurotoxicity that we were focusing on in terms of a readout was the induction of seizure-like activity in these compounds, because we know that we have the GABA component, and the GABA component is very sensitive. So we developed the model, first of all, around this to test an acute seizure-like activity induction due to a chemical impairment of the inhibitory GABA component. So basically, we're dosing with bucuculin, the GABA antagonist, GABA-A antagonist, receptor antagonist, and we can nicely see the seizure-like behavior pattern in terms of increased burst duration, increased network burst frequency, and also hypersynchronization and overall hyperactivity regarding spiking. And so this is an acute model, acute seizure induction model, or seizure-like activity induction model, and we wanted to gain confidence if this is actually representative of a physiological or biological response. And so for this, we applied approved anti-epileptic drugs to see if they can reduce that or counteract these induced activities. And we indeed see, by applying phenytoin and lamotrigine, we can see that in multiple parameters that are deregulated up on the chemical stimulus, we can see a rescue in a dose-dependent manner for these approved anti-epileptic drugs, giving us again confidence that indeed the seizure-like activity that we induce is kind of a biological relevant phenotype. So, and we compared this to gold standard for the in vitro field, or close to a gold standard, not slice cultures, but cortical cultures from rats, and we see pretty much the same pattern. We see the increased activity upon the chemical stimulus in all of the parameters that match the parameters in the human IN assessment, and we also see the rescue of these increased activity upon application of the approved AEDs. However, we already saw that there's at least a difference in the sensitivity compared to human versus the mouse. And so we looked a little bit closer at the difference in the sensitivity from those two models or setups. And we continued testing more AEDs in a similar model using PTX instead of bucuculin, but using a total of 13 approved AEDs for counteracting the acute seizure effect. And yeah, we quantified all the responses at different concentrations and derived EC50 values for each of the parameters describing the neural network activity changes. And then we correlated that to the human plasma levels that are available from databases for those approved AEDs that they're used in clinic. We have nice annotation of data for human plasma levels where this actually this compound would work. And we actually get a very nice correlation between the EC50s on the MEAs and the human plasma levels in real clinical application. It's close to 0.8, 0.9, especially if you take out stiripentol, which partially goes through the GABA pathway and therefore conflicts with our chemical stimulus. And also we did not correct for brain plasma ratios in that specific application. So you would also push the safinamide a little bit up and would probably enhance a little bit more the correlation even further. However, the same compounds on the same applications, if you do the same correlation to the same plasma levels using a standard mouse model for seizures, you already decrease your R-square in that comparison. So either indicating, or probably indicating that there is a species specific advantage using a human iPS-derived neural culture in that. But overall, that gave us a lot of confidence that indeed, going back to the model, that indeed our seizure-like activity model is indeed reflective of something that is biological relevant. And so with that confidence, we started the next test run again in the real world using a set of seizure-genic compounds in clinical use that have seizure liabilities, with the goal to identify parameters that could predict a convulsant risk for these compounds. And so for this round of tests, we were going for multiple modes of actions. So this was covering not only GABA receptors, but also potassium channels, D2, serotonin, and so on. So covering all to get a picture, what can we see, what can't we see with this model. Again, the model is 70-30 plus astrocyte model, which is capable of forming seizures upon disruption of certain pathways. So I'm not going to show you all of the dosing, but just as an example, when we plot all of these parameters that we measure and quantify from the microelectrode arrays recording, we can already see drastic effects for negative control acetaminophen, which is negative in the red bars, picuculin as a positive control on each plate, versus, for example, a compound with a seizure risk, linopyridine, a potassium channel blocker, that shows that specific and well-known increase in network burst frequency, in burst duration, and in firing frequency. So, and dosing all of these compounds and putting all of these compounds in there in different concentration ranges and so on, we end up again with compounds that have specific changes in some parameters and some that don't have changes in parameters or biphasic and so on. So, and if we take all this data collected and make just an unsupervised clustering, we end up basically with two trees, one that basically would be grouped as showing no effect or would not be identified as a potential seizure risk-bearing compound, versus the left branch here showing seizure-related activity patterns or profiles. And very interestingly, is that that nicely correlates with the pharmaco profiling data that we did before to characterize the functionality of those systems. So the potassium channel blockers, as well as I didn't show everything on the pharmaco profiling, but again, be happy to share these data upon request. But we see responses in the pharmaco profiling to serotonin receptors and NMDA receptors, as well as strong responses to GABA A receptors and potassium channel blockers. So again, and we see exactly that. Those compounds that go through these modes of actions can indeed be recognized in terms of being close to a seizure-like activity profile or pattern. Whereas dopamine receptor, we tested in our system, but it doesn't seem to be functionally integrated in that, although it is expressed, at least the D2 receptor. Norepinephrine pathway is not expressed, as well as muscarinic cholinergic receptor pathways is also not expressed in our cells. So that makes completely sense that we don't pick up any of these modulations in terms of a seizure risk prediction. So basically, with the experiment so far, we can already define or start to define an applicability for these assays for a particular mode of action or compound groups to be identifiable in a certain risk range. And again, for D2 receptors, epinephrine modulators, and so on, this assay would be not a fit. So we would either implement dopaminergic neurons or peripheral neurons, something like this. But definitely we have to identify which assay could cover which domain for this tox assessment, and basically then fill the gaps step by step, because again, not a single in vitro assay cannot be capable of assessing all different aspects of potential toxicity. And so we are actually in the process of further refining our applicability by extending the Pharmaco Profiling to more compounds and more isotype specific or isoform specific tool compounds. And the more we know what compounds or what receptor pathways are functionally integrated in our system, the more we can then target and test in training sets and expand our set of those mode of actions that we can actually recognize and identify, to finally get enough data to produce or to generate or identify a signature that could be used as a predictive assay. So this is the overall goal for exactly that application. So that was the last example, and that brings me already to the summary. So just to recapitulate, the Sunfire co-culture system, which again comprises of 70-30 ratio excitatory inhibitory cells plus primary human astrocytes, shows very fast maturation, low variability, and in the QC setting that we have, very specified cell composition as you produce a certain targeted cell population directly by the combination of the transcription factors. And it has a very high level of functionality, many different receptor pathway and signaling pathways that are functionally integrated in a higher order phenotype like a network activity. And this network activity seemed to be, or based on all data that we have, is synapse driven. So synaptic tests can be conducted on these models, and they are responsive to a broad range of pharmacological modification or modulation. And these can be used to identify neuroactive effects of environmental toxicants, new toxicants, but of course only in those domains where we know functional pathways are integrated. And the same holds true for the seizure liability testing. We are in the process of expanding our applicability and the definition of the applicability of our assays. But we're, I think, with in vitro assays, we're on the good way to complement mouse studies and to hopefully soon end up with a panel of in vitro assays that are capable of predicting, robustly and reliably predicting seizure liabilities in vitro. Okay, with this, I think I would conclude. And again, if you have any questions, or if you would like to get some peeks on the data that I presented and some data that I had to leave out for the sake of time, just shoot us an email at inquiries@newsetlabs.com or directly to me. And if you want to test the cells, I guess REPROCELL is your partner, right? Well, and yeah, with this, I would be happy to take any questions.
Thank you, Daniel. If you have any questions, please feel free to use the question and answer feature at the bottom of the screen to submit your questions. But yeah, we have time for a few questions. Do you know what the mixture of neuronal subtypes in your induced neurons is?
The mixture of neuronal subtypes. So we have 70% excitatory and 30 inhibitory cells. So we pretty much define that by the combination of transcription factors. So we always end up with a very homogeneous population, and then we combine it in a ratio that we want to have.
Okay, and how did you come up with a 70-30 ratio for your excitatory to inhibitory neurons?
So that is purely empirical. So we were just informed by literature ranging from 50-50 in some regions, but mostly 80-20 in most cortical regions. So we were simply testing different seeding ratios and tested the robustness of the network development and the responsiveness to AMPA and GABA modulation. And we simply found that the seeding density of, or the seeding ratio of 70:30 gave us the most robust and the most sensitive response to modulation of the inhibitory and excitatory components in the coordination. It has to be said that the ultimate ratio can change over time. So the inhibitory neurons are a little bit more sensitive than the excitatory neurons. So the actual ratio of at the assay, most of the assay time points is actually reflecting an 80-20 approximately.
Okay. Do the anti-epileptics and sedatives that produce effects that block proconvulsant drugs, do these compounds also produce effects on their own at similar doses?
Sorry, can you say that again?
Do the anti-epileptics and sedatives produce effects on their own at the same doses that block proconvulsant drugs?
Oh, you mean if they alternate baseline activity?
Correct.
Actually, so for most of them not. So most of them, they're activity dependent, they don't. But we have actually found some that do reduce the baseline activity. We are currently repeating those data because there were some issues with the dosing scheme, but this data should be available also fairly soon. But yeah, not all of them. Some of them do, some of them don't. But don't ask me in detail for the list. I don't have the corresponding list ready yet. But if you send us an email at inquiries, and can see when that data is completed.
Thank you. How long can the Senfire neurons be maintained in co-culture?
So the standard assay time points are around 4 or 5 weeks, and so depending on what you're looking for, especially if you want to go for extrasynaptic and NMDA receptor currents, you might carry them out a little bit longer. So we have been actually accidentally carrying them out for almost a year, and they were still active and firing. But of course, the variability increases over time when you carry them out, and also media is not so cheap, and maintenance and so on. So I would say I would not extend them past 6 months, but 6 months is, if you have an assay that takes 6 months time point, that I would say is still doable.
Okay.
But you don't want to do that, right? I mean, if you can measure synchronized and mature neural activity after 4 weeks, you want to do that.
Right. So have you tried other medium to high throughput technologies such as calcium imaging with the Senfire neurons?
Yeah, so we have run a pilot project on calcium imaging, and again, this brings out the advantage of this modular producing the different cell types, as you can transduce the reporters in just the neurons or just the glutamatergic neurons, which gets rid of the background noise of the calcium waves of the astrocytes otherwise. And so yeah, we have run in a high throughput setting a couple of dosing experiments similar to the Pharmaco Profiling on the MEAs. And we are currently in the process of evaluating and analyzing and matching that up to the data that we have on the MEAs. And I also think that this data will come online fairly soon, probably this year, yeah.
Have you compared the results from different iPSC donors in terms of how the data you get with inducible neurons compare from different donors?
Yes, at the moment we're just offering the Synfy kit from one donor. We're in the process of establishing the second line with all the QC and so on that's attached to it. But in internal programs and projects, we of course compared IEN cultures and IEN productions between different donors and between lines and also between clones. And compared to stepwise differentiating protocols that in my hands at least were always very variable between different clones from the same donor, we see a very striking overlap in the firing properties and the network properties of these cultures, even between different individuals with that technology. So I think this is a very strong point for using the direct reprogramming approach for assay, for especially high throughput assay approaches and applications.
And are you planning on developing other neuronal type cells such as dopaminergic neurons or astrocytes in the future?
Yes, so there's a lot in the pipeline. Unfortunately, we're not that big so that we cannot prioritize all of the project at the same time. At the moment, we are most advanced with induced astrocytes, and we're running beta tests on the implementation on the platform and the functionality of the developing networks in comparison to the standard culture that I have shown the data from. So I would assume that the next that will come online will be induced astrocytes, and so that we would have isogenic cultures from the same donor, even including the astrocytic component. And other neuronal cell types are indeed in the pipeline, but they will probably take a little bit longer until this is something ready to be given out for testing.
Okay, if there's no more questions, thanks to Dr. Haag for a nice presentation. If you have any questions that come up afterwards, you can email them directly to Dr. Haag at the email address on the slide, or you can email them to me, Bob Anand, through the REPROCELL website, or just reply back to the invitation email that you got when you signed up for the webinar. And thank you all for your attendance, and enjoy the rest of your day.
Thank you very much, Bob.